Hello, my name is Avril and Lindsay. I will be talking about my project Shrouds of Smoke, identifying microscopic low temperature heat alterations of bone in intentional corpse preservation. I did this project at the University of Bradford under the supervision of Dr. Hannah Kuhn. So I know that this is the osteoarchaeology group for CFFA, but I did want to do one trigger warning because I do have an image in here from an ethnographic case study, which is a little bit graphic that does present a picture of fleshed mummified remains that have been dried due to heat desiccation. So if you're not wanting to see that, it's entirely fine if you click off or just turn away for that point in time. So a quick outline of the presentation I'm going to go over. I'll quickly address my aims and objectives, do some contextualization in archaeological and ethnographic studies, then go into the experiment parameters that I used, methods, materials, and then go over my results and conclusions. So the aims of this project was to better understand and detect if a corpse was exposed to low temperature heat treatment prior to burial by analysis of bone collagen and mineral changes. How I went about studying this is I experimentally heated flesh bones to recreate possible past funerary practices, then used Keon's microscope to observe the microscopic changes in structure and color of the bone, then used Raman spectroscopy to investigate chemical changes to the bone, and overall observed and analyzed and then discussed the observed changes in relation to temperature, duration, bone type, and the presence of flesh. So why do this? Of course, expanding on previous research, one of the things I did during my study was a survey of 50 archaeological and forensic studies that looked at heating of bones. A good majority of them in heat studies focus on temperatures above 200 degrees Celsius. That's primarily because there's more interest in looking at cremation studies or partial cremation studies. However, there are factors in which a temperature of a fire or other heating apparatuses might not get above 200 degrees or might not sustain above 200 degrees for too long of time. So I thought it was interesting and important to look at lower temperatures, as well as multiple factors. Many different forensic studies and archaeological studies are unfortunately relegated to being more precise, but therefore less accurate by thinking in a laboratory setting opposed to being more realistic and not thinking beyond temperature. Often things such as duration, type of heating, presence of flesh, burial effects, bone types, and many other factors can be relegated to the side and not thought of whatsoever. Archaeologically, why I wanted to do this was to try to detect theorized but unseen funerary practices. Most primarily was practices in which human remains have been attempted to be preserved, whether that just be for a temporary period of time through heating, through desiccation, drying out of the body so that it can last in a more human presenting form before it is finally placed in a burial. And within that burial, unfortunately, the soil and burial processes just remove the presence of that mummified flesh. By the time archaeologists get to it, there are only hints that something else might have happened, but might also be looked over because you only have the skeletal remains. So some archaeological examples of just that are both in the Near East and South America. Some of these are either confirmed or very likely sites where the bodies were heated at low temperatures and indirect heating to preserve the bodies, whether that be for a few days or for a long procession and period of display before being properly buried. In the South American cases, some of these bodies still have the mummification fleshed remains still present in their burial. And then I also included a few speculative British ones that are based on the tight knit positioning of the skeletal remains that would be conducive to more having flesh when being buried, but in such a tightly flexed manner, it perhaps was mummified, as well as other factors that might have been presented that made archaeologists go pause and say, that looks a little strange. So ethnographic examples, this was the one photo I was talking about that might be a little bit graphic. This photo comes from a study called Beckett and Nelson 2015 of the Anga peoples of Papua New Guinea. In Papua New Guinea, as well as these other documented ethnographic practices, the body soon after death is taken to be presented over top of a fire, never in direct flames, and heated for a prolonged period of time. In the Anga people's case, for at least 30 days, a fire is continuously tended for, in which a body is remained above it in the smoke, so that it can be slowly dried out to present a more humanly or intact form of the body, so funerary rites and traditions can go on for a long period of time before a final burial place is selected. So the experiment parameters I used to try to recreate the conditions of intentional desiccation is I specifically looked at temperatures below 200 degrees, 70 and 120 degrees Celsius, based on modern smokehouse temperatures, as well as experimental archaeology sauna temperatures. I did, of course, dry and indirect heating. I used an oven. And then the durations I chose, based on the resources I was available to have, were 5 and 40 hours. These are directly drawn from experimental archaeology and ethnographic data of earth ovens, which I thought would be a good representative to what a funerary hut might compare to. So the materials I used ovis or sheep bone. It was fresh and intact. I had gone and got it from a butcher that day that I was doing the experiment, or a few days prior, and I chose both rib and long bones because I wanted to both see how different bone types might interact differently to the heating, as well as the different levels and presence of flesh and type of flesh that might surround each of these bones. I chose ovis specifically because in the molecular structuring, particularly in the collagen, more closely resembles in sheep to humans than it does to than pigs, which are often also chosen in experimental archaeology. So my method of Raman spectroscopy, I use this to look at the molecular changes of the bones in the internal structures and ratios. When you're doing Raman, you are shining a laser at your sample, and what's reflect back, it will show these mineral peaks and matrix peaks, matrix being the organic. And these different types of peaks you will base against a control sample, so an unheated sample, and see how it has been altered in the heating process. So the collagen or matrix peaks I was most particularly looked at were the amines and the carbide, and the mineral peaks I was most closely looking at were the phosphates and the carbonates. In the Keon's microscope, I looked at for physical changes to the structure of the bone, such as discoloration, delamination, and cracking. I looked for these around the osteons, lamella, haversian canals, and cortical sheaths. So what were my results? Well, I did indeed find delamination and cracking. However, this was more apparent in ribs. In fact, I believe I only had one long bone sample that had a possible but not convincing evidence for these microscopic changes in the bone structure. I also noted during my experiments that even though similar losses of mass might happen in the ribs to the long bones, if you look at samples 4 and 6, both heated at the highest temperature for the longest amount of hours, they have almost identical losses in mass to their flesh when heating. Yet when I looked at the results in the bones, both microscopically and chemically with the Raman, the ribs were far more affected than the long bones ever were. So there is a clear interest in that bone type is greatly been affected. And further going into the Raman, there was intra-sample variation within the Raman scans. I took 4 points of each Raman sample so that I could see the different changes that might happen and get a cumulative average that I more so assessed between samples. But it is interesting to note that different positions on the bone might be more or less affected to the heating. Rib bones, again, were more greatly affected in their changes to the ramen, more specifically in the ratios and changes to the organic section, likely due to water loss and a slow disintegration of the amnide areas. However, when I was comparing ramen scans, bone type and duration groups showed the most evidence of changes from their baseline samples. So, kind of wrapping up my results and conclusions, delamination and cracking of osteons and cortical bone was likely due to dehydration. If you're trying to desiccate, you're slowly drying it out, so that does make sense and confirms along with other studies that have happened. The difference in heating effects was greatly affected, though, to bone type, which I found very interesting. And the another thing that I had was when I was discussing this with my supervisor, Dr. Kuhn, we wondered if there might be a boiling effect, that the sample might not get above 100 degrees because of the presence of flesh. However, there was not very much of a boiling effect. If there was, it was temporary, a maximum of 2 hours for every single sample, reached its heating. So if I put it to 100 to 20 degrees, we had a thermometer in there and it would reach all the way up to 100 to 20 degrees within maximum 24 hours, if not faster. Duration did have effect on the level of degradation, and in fact, shifts in amnide 1 when exposed to 120 degrees Celsius for 40 hours indicated breakdowns in the protein started to occur prior to 200 degrees Celsius, which was thought to not likely happen, which connects to how duration could possibly mimic higher changes in temperature. So that's kind of changing perspectives of cases of perhaps failed cremation where only the beginnings of discoloration and heating damage present could be signs of more complex funerary rituals, as well as just in general studies how temperature is often the quote-unquote pinnacle thing of, oh, these evidence links to this temperature, where perhaps it might just link to a longer duration at a lower temperature. Future work that I'm going to go do is I will return to the University of Bradford as an outsider researcher, an independent researcher, to do transmission electron microscopy on the demineralized bone samples that I took, and further just investigate the changes to collagen fibers, continuing on work that Dr. Hannah Kuhn has done in previous studies. So that is my references, and thank you for